this morning learning about a lot of very interesting new results that I'll have to think about a lot. I'm going to tell you today about an approach to behavior, brain, and the relation between them. The approach, I would say, is a no-name approach. That is to say, I can't say that this is a particular, zeitgeist or anything like that. And I think it'll stay that way because it seems to me the approach is entirely consistent with conventional views of science. And for me, the conventional views of science mean that if you have a macroscopic behavior, you have to derive it from a microscopic level. Now, it might seem that this is a simple kind of problem, but we already know in the more mature sciences that it's not. So, for example, we have known about the microscopic elements in physics for over 70 years, atoms and so on. Yet, it was only in the late 70s, for example, that we were able to derive physical phenomena such as ferromagnetism and such from a microscopic behavior. That is to say, we previously had phenomenological descriptions of how ferromagnets behaved before we could actually try to derive them from first principles. Likewise, in the case of certain optical devices like a laser, it was only in the mid-70s that it became possible to derive the way lasers behave from quantum mechanical considerations. So these are considered, I think, successes in science, those two things. The theory of ferromagnetic behavior, for example, is called renormalization group. I'm not going to talk about that, except to say that Kenneth Wilson won the Nobel Prize for deriving the behavior of ferromagnets. Introducing this was a controversial Nobel Prize, actually. I could tell you another story, but anyway, all Nobel Prizes probably are. If you accept with me that this micro-macro connection is by no means transparent for the problems we're interested in, then you could ask the question, well, what lessons can we learn from what I've just told you? And one lesson I think we can learn is that it's first very important to know what to derive from the micro level. That is, what is the microscopic behavior of the system that we want to derive? That's crucial. And it's an issue that I think hasn't been appropriately addressed. Secondly, it tells us that even in a system that has homogeneous components, like a physical system, we're talking, our interests here are, for example, the nervous system and behavior and such things, where the component elements are very diverse and heterogeneous and so on. Preferred processing? Laurel? Sorry, Laurel. So you can get these synchronicities that maybe... it tells you really that, in a sense, it's not trivial to derive the macro from the micro in such complex systems. Even, that's to say, even in so-called simple systems like ferromagnets and optical devices, it hasn't been possible to do that in a sort of single step. The techniques are mathematically very cumbersome and so on. So my argument here is that it's not very likely when we come to the nervous system that has hundreds of neurotransmitters and thousands of different kinds of neurons and so on, that we're ever going to be able to do it in a single step either. So to a certain degree, micro and macro are relative terms. What is macro for one person could be micro for another. Nevertheless, I think what the goal would be to see if one can start in a sort of level by level way trying to connect levels of description. That's what I'm going to tell you about. So this, I have to say, by the way, that this work that I'm going to tell you about, I'm the spokesman for it. The two people that have collaborated very strongly with me, usually on Wednesday nights in a local pub, are Armin Fuchs and Victor Yursa. Armin Fuchs, some of the data I will show you comes from his chief interests in computational physics and visualization and graphics. And Victor Yursa is a young theoretical physicist, and it's best if theoretical physicists turn to biology and behavior that you get them young. Here's an outline. Let me take you through this. The goal of this particular approach is to derive behavioral laws from brain mechanisms. I stressed the notion of what to derive. And the general attitude has been to take certain concepts coming from cooperative phenomena and so on, pattern formation in open non-equilibrium systems, etc. Probably people know about that. But these concepts have to do with collective variables, control parameters, and so on. The difficulty we have here is in behavior, trying to identify what the relevant variables are. That's what we mean by a collective variable. A relevant variable that is where wraps up the coordination between the parts of the system. And the idea here is to look specifically in behavior. It's a weakness of this approach, and a strength, in another sense. The idea here is to use transitions as a way to identify what the relevant behavioral variables are. That's to say, in these complex systems, if we are measuring the behavior of a person, that's what we're talking about, it's not at all clear what the relevant variables are. You can measure very, very many things. The idea here, a sort of strategic commitment, if you like, the idea here is let's take this system, this behaving system, to a point where its behavior changes qualitatively. That will allow us to identify what the relevant variables are. That is, those variables that change qualitatively at transitions are going to be the ones that are relevant for the system's behavior when it's behaving smoothly. Okay? So those are the chief variables. There may be many other quantities that we can measure, but the ones that change qualitatively are the ones that are going to tell us about the collective behavior of the system. That's sort of the theoretical commitment. Nevertheless, it has proven possible in quite a large number of cases, using this transition methodology, to come up with equations of motion for the collective variable. Here is a typical equation of motion. Phi dot is a function of phi, and parameters. Lambda here would correspond to control parameters. This is just a prototypical. Don't take this equation too seriously, because for the particular behaviors that people are interested in, the task is really to find what these collective variable dynamics are. And people have done that. People even here have been doing such experiments all over, actually, and for a large number of different systems. Now, it turns out that when you get a description at this level, there are predictions that you can then check having to do with how this equation of motion behaves. You can actually predict different effects that emerge from this kind of dynamics. And these predictions have been checked now in numerous kinds of situations, numerous systems and numerous levels and so on. You now have a description at this collective level, and the goal is to derive it from a level below. So this is just one step down. That's to say, I want to take the components. Here I've just got two components defined by x and x dot. Typical way. This is again, don't take these equations too seriously. The equations exist, of course, but this is just a caricature, a sketch. And the idea then is, can I, by an appropriate nonlinear coupling, derive the collective level? And that too has been done in a number of cases. So here we have a very, if you like, self-consistent description for the behavior of a system. that can be derived from the interactions, the nonlinear interactions among its components. Okay? Such examples exist in terms of, for example, bimanual coordination, multi-limb coordination, multi-frequency coordination, etc., etc. Okay, so now we have a consistent description at the collective level and the interaction among the components. The idea is to use this as a probe into the brain. Okay? So, in fact, again, in certain kinds of brain experiments, and I'll tell you about some of them, or just one or two, very briefly. Again, one has looked for these transitions in brain dynamics and found them. And it's been possible then to identify the collective variables or order parameters for the brain patterns. And these turn out to be spatial modes, spatial patterns that are changing in time. And it's been possible then, and this, I mean, if people want the publications and so on, to again treat these spatial patterns and their time-dependent amplitudes as coupled mode systems and get all the way back to the behavioral collective level. Okay? And all of this really, so here we have a consistent description at the behavioral level derived from the behavioral components and their interactions that's been taken into brain experiments. One has identified new collective variables. Again, one can describe this, the way these collective variables behave in terms of coupled modes. One can get back to this behavioral level. All of this is phenomenological. All of it is just phenomenological in the sense of trying to find out and get as precise a description as possible of the behavior at the overt behavioral level that would be interesting to psychologists and at the brain level, which is interesting to neurophysiologists, for example. And the key point here is, can one now, given these phenomenological descriptions, derive them from, I don't want to say first principles, but derive them from basic principles about what neurons do, how they interact, how they're coupled, what the connectivity in, say, the cerebral cortex is, and so on. And this is just a caricature of the kinds of techniques I'm going to tell you about, where what you see is the scalp. You see a strip of cortex, one-dimensional strip of cortex with its gyri and sulci, and we know that pyramidal cells in cortex are arranged in columns. There's current flow in these pyramidal cells, and that current flow generates a magnetic field that can be detected outside the head, which is a representation of the intracellular dendritic current flow here. And one can now say, given I have a quantity out here that's related to my behavior, it's measured during these behavioral experiments, can I now come up with a description at this so-called neural ensemble level? That is to say that these neurons, in order to produce this 200 femtotesla signal, you need a chunk of cortex of about 0.1 millimeter cubed of densely packed neurons of around 10,000 neurons simultaneously active in order to produce the necessary current to be picked up outside here with one of the most sensitive detectors that man and humans have developed called SQUIDs, superconducting quantum computers. And this is called magnetoencephalography. So what I'm going to try to do is give you the flavor of specifying a theoretical description at this neural ensemble level that will take us right back to the phenomenological description of the brain, to the components, and to the collective behavioral level that it took about 10 or 12 years to describe. That's the talk. I was going to make some parenthetic remarks about this this collective variable. I'm going to show you one just specific example of the behavioral dynamics, and it's a specific example then that it has been possible to derive from this neural ensemble level. But I want to make it very clear that this is just a prototypical dynamics, and that each situation has to be studied on its own level and in its own terms. So for example, if you go to trajectory formation or something like that, you can't explain how people produce trajectories of multi-joint limbs by just a phase description. You need to include the amplitudes and so on. And you need to include the perceptual pickup of the trajectory. It's necessary to do these kinds of things. Or if you are interested in what people call anchoring effects, you need to consider anchoring effects are that when you're doing, for example, bimanual coordination, you don't see these typical bimanual transitions, or they're delayed, and the reason seems to be that the metronomes that are specifying the endpoints of these trajectories are helping the system stabilize. That, again, has to be understood on its own terms. Now, at the same time, the argument here is that there are likely to be only very specific windows into this approach. It's not that this is a panacea for everything or anything like that. I made the point that the approach is especially strong near transition points. Not everything exhibits transitions and so on and so forth. So, for me, part of the philosophy of this approach is inventing or discovering experimental model systems that one can actually identify some of these features. For me, for example, I'm interested in certain general biological features of behavior. For example, the fact that all behaviors are synergized, that they're stable, that there's a selection process for behavior going on, that they're multifunctional, I can do different trajectories and still accomplish the same goal, and so on. Those are the kinds of general aspects I'm interested in, and I want a coordination dynamics, I want equations of motion that will capture those kinds of phenomena. So let me just tell you about a specific experiment, show you the behavioral dynamics, and then I'll go to the brain. So this is a very simple experiment, but it shows you just the kind of thing one is going to examine here. There's a stimulus, so-called stimulus, and the subject's task is to syncopate with the stimulus. That means the subject has to produce a movement in between the stimuli. And the stimuli frequency of the stimulus is going to be increased as a control parameter. Control parameter here means I'm going to turn the knob every 10 cycles and make this frequency go faster, and I'm going to ask the subject to stay syncopated. Non-trivial, actually. It's quite a nice little task. But what has been observed in these experiments is that people can only syncopate up to a range of frequencies, and then there's a kind of transition, and then they go, as you can see, this red line here is the point where they switch to a synchronization strategy. So as you change a parameter going from left to right with time, there's a switch from syncopation through a transition regime into synchronization. The key point one should realize here is if we started the system in the initial conditions of synchronization, it wouldn't shift at all. So theoretically that means that syncopation and synchronization are both possible, both behaviors for the same parameter values, but as this parameter increases, only syncopation will switch to synchronization and you'll get a transition. And you can see this kind of effect very nicely if you plot the phase relation between the stimulus and the response. I want to say something just very briefly about this notion of stimulus and response. Syncopation seems to require you to know something about the stimuli and the interval of time between the stimuli so that you can put your movement in between these stimuli. Synchronization, however, means that you actually put your response before the stimulus. There's a degree of anticipation involved in both of these tasks. So it's not strictly speaking sort of stimulus response. But in any case, so plateau means here that for 10 cycles, the frequency is at 1 hertz, then it's increased systematically 1.25, 1.5. And what you see here is around 2, 2.25. In this particular run, this is just one run of the experiment. You see that the relative phase is shifting slightly from 180 degrees. Then you get this phase wrapping kind of effect, this kind of transient effect, and then it switches to synchronization. Okay? And that synchronization is stable. So the modeling step now is to accommodate this kind of effect. And this is just one system. There are many other systems that exhibit the same kind of stuff. So let me just briefly show you the characteristics of the behavioral dynamics in this situation. This is probably familiar to a lot of people. As I say, the same kind of equation has been studied in a wide variety of paradigms, including by Paul and others, Jeff here and many others. But this is just a little pedagogy in this. The phase relationship here is the collective variable. That's the variable that changed qualitatively or abruptly. It has time dependence. So this is phi dot is just the derivative of the phase, which is some function of parameters. And the parameters here that are relevant, you can introduce this first term, which has to do with the frequency differences between the components. If you want to operationalize that in this experiment, it's the frequency difference between the metronome and the preferred frequency of the hand, or something like that. I prefer to see this delta omega in general as a term that breaks the symmetry of these dynamics. Because you've got bistability and a bistable to monostable transition, you need to have this sine 2 phi term. Maybe Paul talked about this a little bit on Friday or something like it, not really. Okay, so, so this is kind of well known, that you can consider these lambdas here as coupling parameters that are related, for example, in the experiment to the frequency that you're driving the system. And all real systems contain fluctuations, and this is a term that says the fluctuations here are just stochastic fluctuations, delta correlated, there's no memory in them, they have a noise strength Q, and so on. And this kind of equation, everything's been quantified very nicely over the years with specific experiments. The way this equation behaves, I think, is really the interesting thing. That's to say, the way to look at this is to look at phi dot against phi, a phase plane trajectory, or as a vector field. And where phi dot crosses the x-axis with a negative slope, this means that you've got a stable state. Okay? So here is the regime for certain values of the control parameters or coupling parameters, that you've got both a synchronized or a nearly synchronized state with a small phase shift due to this asymmetry, near 0 and near pi. The unstable states that surround these stable fixed points are where the slope of this function through 0 is positive. So these are called repellers, right? These are repelling states. And what happens here is that one point here is that for the same values of the parameters, you've got two possibilities depending on how you compare the system. So two things. The system is multifunctional in this sense. That if you now change the parameters, what happens then is there's a bifurcation. The state around pi disappears, leaving the state near 0. But you can also see that that has shifted. The mean position of that has shifted. And you can see the reason why it shifted. The reason why it shifted is that this bifurcation is called a saddle node or tangent bifurcation. And the reason that happens is that the stable fixed point near pi and the unstable fixed point have collided. The function's lifted off the line, and there's no longer any stable fixed points. There's no longer any fixed points in the system at all, except the one for phi around 0. And then as you change these parameters further, corresponding to the frequency of the experiment, you see that actually the system is no longer showing you any coordination at all, except it's showing you actually that there are remnants of the previous coordination states. See? And I think this is a very interesting situation. What it means essentially is that while there are no stable phase-locked coordinations, there is a kind of a tendency to show coordination. And that tendency is realized by components that are trying to express their own individual variation. So this particular regime I'm very attracted to as a regime where you're no longer mode locked, but you're not stochastic or chaotic either. There's still a partial coordination going on. It tells me, or says to me, look, this is a way we can reconcile the need for sort of integration while allowing the parts to be express their own individual variation.
Do you see any sign of that intermittency?
Yeah, you do. In fact, it's been quantified in experiments. There are particular predictions about how close to the fixed points you are, and how the dwell times around those fixed points should change.
Yeah, is it fitting in one of the classic types?
It's type 1 intermittency, so-called type 1 intermittency. Okay. And as I mentioned, that particular dynamics, I mean, this is an old slide, and it's much work has been done since this, but the same dynamics handles the motions between components of the same system. The description I just described to you between a light and a sound and a finger in terms of coordination between the organism and the environment, beautiful experiments between organisms and so on. Okay, so my idea here is to try to use this now as a probe into the brain. And I'm going to just describe one experiment, which I'm really excited about. But this is kind of just to show you the setup. Over the years, we've done experiments where we have 36, 37 squids over the left hemisphere. The newer work, the work that I'm going to describe to you today, has 68 squids over the entire head. So you get a full head coverage. And the technology now, and this is done in collaboration with CTF Systems in Vancouver, who are one of the few places in the world to actually develop this technology. They're now up to approaching 200 on the head. And of course the idea is that you get very nice temporal resolution, millisecond by millisecond temporal resolution, but you can also, using particular techniques, start to get much improved spatial resolution as well. The limits of that technology are nowhere near in sight. But you can see what we're going to do here, or what we've done in the past, not what I'm going to tell you about today, where we've done this syncopation, instability, synchronization experiment and seen all these dynamical effects, and it's been published and so forth. I think I need to say much more about the technique itself. Here we come back to this picture. The generation of the signal is through intracellular current flows in these apical dendrites and pyramidal cells. Of course, you're only going to be able to pick up the magnetic field tangential to this current flow. The benefits of MEG over EEG are several. The magnetic field goes right through the skull, and the interstitial tissues is picked up as a pure quantity. The EEG is picked up as a potential difference due to extracellular current flow. And that EEG, although techniques are improving there, brain atlas, whatever, nevertheless has the problem of volume conduction, and it needs to be considered. But people are using a lot of different techniques to try to overcome that.
We couldn't afford a squid.
Right?
Exactly. We don't afford squid.
That's the problem with the brain. Nobody can. That's why you have to write Okay. In this, I'm going to tell you about, I think, a very interesting little psychological effect. In the original syncopation synchronization experiments, there was one subject who didn't switch. In fact, one subject who failed to show any effects on syncopation at all. That is, she was started with syncopation, but she always kept syncopation right up to 4 times a second, 4 beats a second, 4 hertz. And on asking her why she was able to do this, you know, what the hell was going on, she said, well, you told me in this experiment to make a flexion movement off the beat. Right? So the person's making The beats are coming boop, 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 and they're going to go faster, and I've got to make a flexion movement off the beat. She said, what I did was I extended my finger on the beat. I synchronized on the beat, and then my flexion movement would automatically go off the beat. Jeff's so interested in cognitive strategies and so on. This is one of beautiful cognitive strategy on these dynamics. She treated the task as a synchronization task, but she accomplished the initial goal of the stupid experimenter of perfect syncopation. So I wanted to look at this, and here's an experiment where subjects are doing all four conditions. They are going to be flexing on the beat or extending on the beat, flexing off the beat or extending off the beat. And so what you're seeing here is the result of the trajectory, which is on a pressure cushion that you can pick up as movement trajectory. And you can see this is the resulting phase. People in the synchronization conditions can do that very nicely. They're around 0. This is just repeated in the interval 0 to 2 pi or 2 minus 2 pi radians. They're pretty good. They're doing this for 100 cycles just at 1 hertz. No manipulation of control parameter. Long-term stuff. Because we have to get the EEG or the MEG signals appropriately. We have to get an appropriate sampling to get nice results. That's one reason. But you can see already even at 1 hertz, t equals 0 is the peak of the movement for these different tasks. And what you're seeing here is an array, I think, of about 7 snapshots out of 256 in 1 second. Okay? Remember, this is a 1 hertz movement. And what you see is something I think quite interesting. That, let's take one here, that, say, in this flexion on condition, there's not really much structure here, but at a certain point in the movement, you see this dipolar structure come up, and then actually it disappears in the other phase of the movement. Likewise, for extension on, you see the dipolar structure come on again at a particular point in the movement, and then either weaken or reverse itself in the second phase of the movement, and so on. But the main thing here, and I think it's seen quite nicely here, is that regardless of whether you're flexing on the beat or extending on the beat, you see this dipolar-like structure that's associated with the movement itself. Okay? You can see it here for flexion off, here for extension off, here for extension on. Same thing, right? I think I'll miss the next one, but well, I can explain it. Here's the actual magnetic field pattern, the time series of the field pattern. And down here is plotted the movement itself and its derivative. Okay? That is the velocity of the movement. And what I'd like everybody to sort of ask themselves in this task of synchronization or syncopation, how do you actually do it? How do you actually get your movement at its maximum amplitude at a particular point in time? Okay? And what you see here is in a number of sensors that the brain signal in red is superimposed on the derivative of the movement. Okay? And this is just for one condition, the flexion on condition. But actually, as I'll show you, it actually is the same for all the conditions. So you can see that this velocity of movement is correlated almost one with in a certain area, namely if the nose is up here in the left sensory motor cortical area. Right? So it's either correlated with with a positive correlation coefficient, or it's anti-correlated because in one area the brain is exiting, the field is exiting the head, and the other it's entering the head.
Are we looking at the average of 22 subjects,
No.
1 second each?
No, you're looking at the grand average of 5 subjects over a 1 second period. So, and in this case, there are 228 1 second chunks in that average. Okay. Well, one of the things one can do with the brain patterns is you can say, given this brain pattern, can I compress that information in some way that will be meaningful? And a way that people have used it, or looked at this, fluid dynamics and other areas, is that they have done what some people call a Korhunen-Lev expansion, or a singular value decomposition, or principal component analysis, and so on. That's to say, what they're asking here is, given I have a spatial pattern produced by 68 sensors, can I actually see just a few spatial patterns that characterize that entire 68-dimensional array? And that, is part of our analysis. And what I'm going to show you is that, well, in area work, from this kind of work, you can actually do that. That's to say, these lambdas here are the eigenvalues of a covariance matrix. The spatial patterns themselves are the eigenvectors, and you can actually get a value for the contribution of, say, this spatial pattern to the entire signal. And you can see that this first spatial pattern accommodates 59%, the second 19%, the third 8%, and so on. Often in these kinds of situations, with just 2 or 3 or 4 spatial patterns, you can accommodate about 95% of the variance in the data. And of course, this is a singular value decomposition. This is a linear technique. These are orthogonal spatial patterns, and so on. There's been quite a lot of work, changing the basis vectors and doing non-linear analysis and so on. I won't go into that, but it's part of the bag of tricks. When you do the spatial pattern analysis, guess what? The top spatial mode, which is under no orthogonality assumptions at all, right, is precisely the one we saw when I showed you these snapshots of the spatial pattern. It's this one here. It accommodates actually about 50 to 60% of the entire variance in the signal. And what you can do here is you can take the time-dependent amplitude of this spatial mode and look at its relation to variables that are seen in the experiment, in the behavioral experiment. And what you see is that the time-dependent amplitude of this spatial mode tracks, this is the overall, tracks the velocity of the signal very well, independent of whether you're flexing or extending, or independent of whether you're synchronizing or syncopating. And these are the individual conditions. Here I've plotted the derivative down here, and the brain signal, and you can see it's beautiful, that the brain signal is tracking the derivative of the movement very nicely indeed. I can show you that in a slightly different way. Here is basically the same data, and I'm showing here on the left just the first 2 spatial modes. And with 2 spatial modes, you can accommodate about 80% of the data. But the key point here is that this top spatial mode, which is really, I think, physiologically significant, that is to say, it gives you this strong dipolar structure in the left sensory-motor cortex, is producing a signal that's tracking the derivative of the movement beautifully. Now, our reviewers said this is very interesting because you've shown here that the brain, seen as this sort of population activity in the brain with this technology, is tracking the derivative of the movement independent of the direction of whether whether you're flexing or extending, and independent of whether you're syncopating or synchronizing. This is really a movement or a movement-related effect. Does it change as you change the velocity? So, fortunately, when I was at CTF, I'd done an experiment in which we changed the frequency. It's not a perfect experiment, but here again is the top dipolar mode, and it's projected, its time-dependent amplitude is projected, and you're onto the data, and you're seeing a correlation here between the derivative and the time dependence of this spatial pattern, and you can see that it follows it very well, especially in the initial active phase of the movement, less well in the more passive phase, which we think is probably related to the sensory component, and so on. So you can see that that's there, regardless of what frequency you're using. And
which color is
which color is which?
Yes.
The red is the time-dependent amplitude of the spatial mode, and the blue is the derivative. How am I doing the time? Oh my goodness. Okay. All right. Okay. I promised you that I would try to. So here let me summarize. Here we have a signal coming out of the human brain that is corresponding to the derivative of the movement that's task independent and direction independent. And it's, I'll show you right at the very end here that people have seen some very similar things in single cells. I wanted to now just discuss the neural theory of this a little bit. Let me see if I can do this. It's working here. Here is the picture I showed you before. And what I want to try to do is kind of effectively zoom on this picture. The signal is generated by current flow in these apical dendrites. That current flow, of course, is going to be produced by cells, pyramidal cells in the cortex, and the neurophysiology shows that these apical and basal dendrites extend right through 6 layers. So this cortical column has 6 components to it. There are synaptic connections. I don't like these words of output and input, but just to convey the notion that there are projections out of these that cross the white matter into other areas of cortex, and likewise there are inputs into this cortex from other areas as well. But what I've really tried to schematize in this caricature here is that the magnetic field is generated by intracellular current. That intracellular current is in turn produced by cells here that have particular conversion operations. That is to say, at synapses here along this axon, you can see that pulses, action potentials, are converted to currents at synapses. And likewise, at the cell body itself, it's the opposite. That's to say, currents build up to some threshold and produce action potentials. Okay? Those are 2 well-established neurophysiological principles of synaptic transmission and the generation of action potentials in cell bodies, trigger zones. Now, the theory is not going to talk yet about this single cell level, but what we're going to do, because there are well-known effects, what we're going to do is zoom the other way and consider this piece of cortex as a one-dimensional strip here. Let me, for the sake of visualization, pull away this. And let me just do that for a moment. So we're now going to envisage, does that do it? Yes. We're now going to envisage this piece of cortex as a strip where the neural activity psi, it's changing in space and time, has got excitatory and inhibitory neurons that have intracortical connections, that is within a particular column, and also cortical connections. Okay? So there are excitatory connections from one area of cortex to the other, and there are also excitatory, and inhibitory connections within a given area of cortex. And those ensemble operations are governed really by the same kinds of conversion operations, now seen as a population activity that we saw for the single neurons. That is, from pulses from action potentials to currents, the conversion operation is sigmoidal, but it's confined to a linear region. At the cell body, this sigmoidal function is there as well. Currents generate pulses according to this sigmoidal function. So that's the sort of architecture of the model I'm going to talk about. Let me reinforce that with some words, that we're talking about excitatory and inhibitory neural ensembles. There are connectivity constraints, both intracortically, where the cells have both excitatory and inhibitory connections. The spatial ranges here that are considered are of the order of a millimeter and a time scale of 1 millisecond. This is all pulled from known neurophysiology and neuroanatomy. There are likewise cortical fibers that are known to be predominantly excitatory. Like 90% of connections from one area of cortex to the other are excitatory. And long range, in the order of 1 to 20 centimeters, with time scales of the order of 20 to 200 milliseconds. And the variable we are measuring here is this psi in space and time. As I mentioned, this dendritic current flow in space and time has an intracellular source for the MEG and is extracellular for the EEG. And what Victor Ursa has done here from these basic neurophysiological constraints has come up with a nonlinear partial differential equation for this variable psi, that is the measured variable from the MEG. This can really be connected to really physically measurable properties. It's a second order differential equation. Don't be so concerned about the terms here too much. But on the left-hand side here, you see, it's sort of Newtonian in a sense. Psi double dot plus psi dot plus psi equals then something here on the right-hand side where this S corresponds to the sigmoid function and these P's are inputs and outputs into this strip of cortex. So omega is a characteristic frequency connected to the axonal propagation velocity, V over psi, or sigma, where sigma is the mean cortical fiber length. This is Laplacian, which governs adjacent neighbor interactions in this cortical sheet. The S is the sigmoid function that I showed you, the conversion operation, which is a consequence of the summation of threshold properties at cell bodies. And in our experiment, we have inputs to this neocortex and action potential densities. We don't talk about individual action potentials at this scale, but only summated or densities of action potentials seen here in terms of afferent fibers to the cortical strip. Now, this is the way it looks in terms of our experiment. And I think this is the conceptual connection that I'm trying to convey. I showed you the behavioral level here of a visual stimulus coupled to a finger movement. And I showed you what that behavioral dynamics was, behavioral level. Now when we look at this in relation to levels of observation of the brain, we see this visual stimulus, again using the term input here, that is going to activate a region on this one-dimensional cortical sheet, this beta region. This is the activated functional unit of cortex, which is specialized for receiving this visual stimulus. There are intercortical connections here, and cortical connections between visual and motor areas. And here you see the functional unit that's activated, generating the finger movement output. And likewise, in a closely related way, there is sensory input or sensory feedback from the finger movement. Okay? So this is just a way to see the structure of the theory that's corresponding to the experiment in this case. This behavioral level, if we were talking about bimanual coordination, would have left finger, right finger, and so on. And that's a different case. So when you look at this, what you want to see here, this is just a little theory, very quick, where this size of t is again our time-dependent activity of this measured variable, this dendritic current variable picked up by the magnetic field. This betas of x is the activated functional unit. And what you're seeing here, and that activated functional unit here is this dipolar structure that we saw in experiment. And what this really amounts to is an integration in time and space, where this betas of x represents the motor area that we saw was activated in the experiment. We assume the finger movement is linearly driven by the output of this brain signal. And here is our model for the finger movement. It's an oscillation, of course, z double dot plus z. It has a damping coefficient that must be positive. Now equals the corresponding generating field, the generating activity that's acting on this finger movement. If you do analysis on this, you can actually drop the second order term for the motion itself, and you come up with this, and the solution of this actually creates the desired phase shift between the movement and the neural signal. That is the velocity coupling. And here is the results of that. So when you integrate this model and project its time-dependent behavior, use that to drive the finger oscillator, what you see here is here's our spatial pattern again, dipolar structure, and here is it driving this finger oscillator. The blue here is the movement, okay, the actual original signal, and the red is the reconstructed signal. That is the result of our theory. And you can see actually it's very beautiful that the theory produces this effect for all the conditions: flexion on, extension on, extension on. Especially as we saw in this initial active phase, it's less good out here where we think there's sort of sensory components. When you look at that as a rhythmic behavior, here's what you see. Here, red is the time-dependent amplitude of the spatial pattern. Blue is the movement. Here's with noise. And you can see that there's a phase shift between the brain signal and the oscillatory movement. That would mean that it would be perfectly superimposed on the velocity. This is a velocity-coupled system. And here's just one neural pulse in relation to the movement, and you can see the neural pulse, the brain signal and the movement signal with the phase lag. Now, it turns out, I mentioned Georgopoulos' work, but Andy Schwartz at the Neurosciences Institute has done experiments in the same paradigm as Georgopoulos, except what he did was, instead of looking at the population responses of cells that are direction specific, he removed the direction effect. He just averaged across all directions, and he looked at the relationship between the firing frequency of the cells and the velocity of the movement. This is now monkey single cell recordings from monkeys, and this paper is about to appear, but I should have color coded this, but the right trajectory here, this one, is the movement, and this is the population response of the cells. So when you remove the direction component in the same cells, you find what they call a neural speed component that's intimately connected to the movement itself. Just exactly what we saw in the human brain. And it's the same cells producing that show a directional effect are also producing this effect. The cell is multifunctional. Same part of the cell is multifunctional. So I'm very excited about this because, A, I showed you an experiment, a very simple experiment, that showed that independent of the direction of movement, the brain activity was strongly correlated to the velocity of the movement. Makes a great deal of sense if you ask yourself, how do I synchronize with an external event, which everybody in this room can do? That means I have to regulate my velocity so that I arrive at the right place at the right time. I showed you that that is in general independent of whether you're synchronizing or syncopating. And it seems to me that it's a very nice and neurophysiological or a brain level of description for a kind of motor equivalence. So just to say at the end, what I tried to do here was using various concepts of ordered parameters or collected variables control, a whole methodological assumptions about how to do experiments. I showed you a particular behavioral experiment where the dynamics have been identified. The phenomenological description of those dynamics at the brain level has been described in terms of these coupled spatial patterns which act as order parameters with time-dependent amplitudes. And I showed you the very beginnings of a neural theory that can actually take us back from the level of how the neural ensembles interact with each other in the cerebral cortex that are generating actions in the environment, how this level can then be connected right back to the actual behavior. And I've tried to show you that if you look at the data the right way, and if you have a certain kind of experiment, you can make this connection a very intimate one.
Questions, please.
In the task you described, I noticed when you were showing us the graphic model, you had visual input.
Yes.
Does that mean that what they were synchronizing to was a visual representation?
In this particular case, sorry, can you explain that? In this particular case, it was a visual metronome, a flashy metronome in front of them.
So what we're seeing in those maps is using terminal.
Yeah.
Is a
Well, there are orthogonal, you see.
There's a time lock, a time relationship, a temporal relationship between a stimulus and a response, an output.
Yes.
That's in the area we would expect to see for the right-hand finger, correct? Finger of the right hand, correct. What we're not seeing in that map, though, is any time lock to visual processing. How come we're not seeing a time locked visual generative potential in occipital temporal cortex?
That's fine. What you are seeing here is the largest activity. Okay, you noticed, or you may have noticed, that for each one of these experimental conditions, they were individually scaled for the largest amplitude of activity. This doesn't mean that there hasn't been activation in visual areas or auditory areas. It just means that the largest activation showing this dipolar structure is movement related. That's very clear.
Because in terms of establishing the brain behavior relationship,
Yes.
it would seem that you need to do the same experiment or the same analysis, showing a time-lock relationship between the visual stimulus
That's no problem.
and the motor output and the visual process.
Yeah, that is no problem in terms of standard evoked field technology. That you have visual input and you get a visual response. What is important here is that this is a synchronization, syncopation behavior. And that task relationship swamps the response that you see in the...
Just like with the EG.
Yeah. Actually, it swamps it. In the EEG, you wouldn't see it really at all. The MEG resolves these spatial patterns much better, actually.
You wouldn't pick anything up with the registration of the visual or the auditory stimulus anyway.
No,
It's the same, it would be just a constant through the whole.
Well, no, but the point is that since you are plotting a relationship to the velocity, right, then that same time-lock relationship will exist with the visual stimulus as well.
Yes, but it's...
Whichever point you select,
Yes.
you should show visual processing as time-locked.
In previous work, and I don't want to show you that, I've got some stuff here, but actually, when you do a Fourier transform of a brain signal, you will pick up a very strong component related to the stimulus frequency. Okay? The point is that when you look at this just in relation to the movement itself, that is, you're averaging these data with respect to the movement, you see this very strong dipolar structure. And of course, what's interesting is its profile. It's the profile that's really interesting here in relation to what people are doing. It's a velocity profile and the profile of this time-dependent dipolar structure.
When you adjust the scale,
Yeah.
do you actually find anything happening or any potentials related to supplementary or premotor areas? Because grossly, that looks like it's in motor cortex.
Yeah, you see, again, when you do this principal components analysis, and I'm just showing you the top principal component, which is quite nice because that's only 60% of the variance in the total signal, but it's picking up the largest activity pattern, which is this dipolar structure. Other spatial modes could be picking up other things, but that's not what I'm trying to do here with this.
Well, the other thing which I was wondering about was how generic is this spatial pattern? Suppose you're asking people to tap their big toe or their left shoulder or something like that.
Yes.
You get the same spatial pattern, or is it a little different area?
That's a good question. I don't know the answer. I don't know the answer. You know, I would say that these kinds of experiments take a long time to analyze. And so to do an experiment where you would say, move a different part of the body, shoulder or elbow, would you see the same kind of dipolar structure? That would take some time and effort. Might be able to do it. I can't say anything. All I can say is that that same dipolar structure is independent whether you're moving, flexing or extending, or the task or the variable itself.
Could you comment on a general concern of one of the concerns I have about the high-tech analysis system is that we sometimes forget that we're not actually looking at the actual microbehavior. We're looking at a representation of microbehavior filtered through algorithms which simplify what the computer does. And it concerns me that when we go to high-level mathematical modeling, what we may be modeling is the mathematics of the analysis, not the mathematics of the system.
I agree with you. I agree with you. In this particular case, and in fact, the phenomenological description of the coupled modes was like that. That is to say, I could not attach any particular physiological significance to these principal components. But in this case, because it is the dominant principal component and has this dipolar structure, and the person's making a right-handed movement of a particular velocity, I think I can claim that this mathematical technique, which is not violating any orthogonality assumptions at all, it's a top principal component, just like the neural ensembles are not violating any mathematical principles, but in this case they're connected. And so that's what I would say is a singularly exciting aspect of the mathematical analysis in this case.
It's going to complicate analysis of the other components.
The other components I can't say very much about brain at all, except to say that you only need 2 or 3 of them to accommodate about 95%